All right, so just recapping the gel electrophoresis, the steps of doing it. You first have to create a gel made out of agar solution, carbohydrates. You then put a specific buffer solution into the electrophoresis tray. You put the gel in, and there's little divots in that gel, and you add your protein samples along with your standard sample or your known sample. You then apply the charge or the current, and the protein samples will move along that current. Based off of size, the larger ones will travel slower and the smaller ones will travel further. You then have to spray the sample with a ninhydrin solution or other dye that will adhere to proteins, specifically amino acids, and you then compare the distance that the various proteins traveled with your standard or known sample. And that is gel electrophoresis in a nutshell. Yeah, this is page 690 of the Pearson book. I'm trying to zoom up on the question. It's kind of blurry. And why don't we go right, did I say number 7? Yeah. So number 7 is saying consult section 33 of the IB data booklet. I got a pink version of that right here. And I think the numbers are slightly off. Oh, here it is, page 33. Because on my booklet it says page 31.
Yeah, it's just section 33.
Oh, there it is, section 33. Got it. Okay, so it's not off. Page 31, page 32, that has all the different amino acids. It has their isoelectric point. And the isoelectric point is where it's a Zwitter ion, so it's the pH that it's going to be a Zwitter ion, which means it has a positive and negative charge at the same time. So it says, consider the following amino acids: glutamine, glutamic acid, threonine, lysine, and serine. Which of these amino acids is most likely to be found? I want to start with C and D. Will be negatively charged at pH of 5. Why don't we just look at leucine first, which is right here, and it's a pH of 5. So right now it's well, it doesn't have anything. It should have a plus and a minus charge if its pH is 6. But then we got to think, if our pH is lower than 6, is that going to be a high H plus environment or a low H plus environment if we have a pH lower than 6? So what does it mean when you have a low pH?
It's acidic.
It's acidic. Therefore you have a high H plus environment. So if you have lots and lots of H pluses, let's see, do I have a pen? Alright, we got H plus, H plus. Why don't we just draw this? We got H2N. So we got tons of H pluses. What's going to happen? Is this amino acid going to gain H's, or is it going to want to give away H's if we're thinking about equilibrium?
Give away? Wait.
Well, where is the concentration highest for H plus? In the environment or in the amino acid?
I don't know.
So in the environment, since it's a low pH, it's a high H plus environment, we got all sorts of H pluses. And then in the amino acid, comparatively, it's a low H plus environment. So equilibrium, we're going to go from high to low so that it stabilizes or equalizes. So that means the H pluses are going to go where it can be added, which is right here. And if we rewrite it, we've got H3N plus. You're going to have a couple of these on the test where it asks you which direction is it going to go. Is it going to go towards the positive end of the electrode or the negative end? And it's kind of tricky. It takes some thought. So low pH, you got a high H plus environment. You got H pluses that are going to then go to attach to the amino acid so that the environments kind of equalize. And that's going to create a positive charge for this amino acid. So, going back to the question, we have a pH of 5, will be negatively charged at pH of 5. Alright, so this type of situation for leucine is going to be happening if you have a pH of less than 6. So we're currently in a pH environment of 5, which is less than 6, but that creates an image like this, which is, well, it's positively charged. Then we got to think of the question, oh, will be negatively charged. So what we've identified here is this is not the amino acid that we're looking for in question C, because it's going to be positively charged, not negatively charged. So then you'd have to do the same thing for all of them until we finally find one that's negatively charged at pH of 5. So let's do another one. Glutamic acid is right. Got to zoom out. Let me just zoom out here.
I don't think we're seeing that.
Oh, oh my gosh, what a waste. Oh, gotta share. Okay, thanks for letting me know. Oh, I gotta click on it. I clicked on it, but I didn't click on it a second time. There we go. Oh my gosh. Oh, what a waste. All right. Well, we got another example. Oh,
that's much.
That's why you. That one. Oh my gosh. See, so this is a high H plus environment, and this is a low. So we're going to add H's to the NH2, and it's going to form NH3 plus. So it creates a positive charge, and this happens when it is a pH of less than 6. A low pH environment, it's going to be positive. But the question was asking, is it negative at a pH of 5? Our answer is no, it's not. So that's not going to be our answer, leucine. So now we're going to look at glutamic acid. All right, another sheet of paper. Glutamic acid. So glutamic acid is right here. Well, I guess I'll just write the other parts. And it's not too important to have to write them, but so right here, 3.2 is the isoelectric point. That's when it's going to have a positive and a negative charge, and we're looking at a pH of 5. So pH of 5, relatively speaking, would be considered a low or high H plus environment, if 3.2 is kind of the cutoff. So anything below 3.2 is this, above 3.2 is that. I'll give you a hint.
Probably like a lower H plus?
It's going to be a lower, yep. So if it's less than 3.2, a high H plus environment, because it's acidic. And if it's higher than 3.2, it's going to be a lower H plus environment. So I'm just going to write 0 H pluses. And you're not going to be asked to visualize. I'm just showing this as a visual to help you kind of figure it out. So low H plus, so I'm not going to draw any H pluses. That means the H pluses are going to be lost from our amino acid. Do we take them from the amine or do we take them from the carboxylic acid? We take them from the carboxylic acid. So this would then be something like this. And if it's extremely low H plus environment, then you'd lose the other H also. But the main idea, it forms a negative charge if it's a pH of 5, and this is glutamic acid. Alright, and the thing is, for C it said will be negatively charged at pH of 5. So this, anything above 3.2, is going to form a structure like this, which is negative. So for letter C, glutamic acid is the one that's going to form a negative charge at a pH of 5. Alright. You guys want to do another example?
Yeah.
Alright, let's do another example. Because we still got to answer D, which is positively charged at pH of 7. This one was leucine, and it was positively charged. Let's just do a different one. So one of the other ones, sharing your screen, 309. So we got 309. Alright, so 309 is right here. I'm just going to write writing that just because we got the amine, we got the carboxylic acid. Every single amino acid has that. That's the main idea. 309 has an isoelectric point of 5.6. So we're looking at pH of 7. So we got to figure out 5.6. Anything less than 5.6 is going to be a high H plus environment. Anything more than 5.6 is a low H plus environment. Alright, and since pH of 7 is above 5.6, we're going to be working with a low H plus environment. So what charge is going to form on this amino acid? Just generic amino acid. Lysine 9.7 is the isoelectric point. So that means any high H plus environment is going to be less than 9.7. Because the question is pH of 7 on this one, so positively charged at pH of 7. And 7 is going to be less than 9.7, so that's in the high H plus environment, because the lower the pH, the higher the H plus. And so that means we have our amino acid, we have our environment. Which direction is the H plus going to be going? Into the environment or into the amino acid during a high H plus environment?
Amino acid.
Into the amino acid, you got it. So that means it's going to attach to the amine, and we're going to have H3N plus CE or COOH. So at a pH of 7, we're going to create a positively charged amino acid that actually happens to answer our question, will be positively charged at pH of 7, and this was lysine. So lysine is going to be positively charged at pH of 7. If we look at what if it was a pH of 11? pH 11, this is now a low H plus environment. So therefore the H's are going to leave the amino acid into the environment. So that goes away and it forms a negative charge. So in a low H plus environment or a pH greater than 9.7, you're going to create a negatively charged amino acid. Alright. And if it's at the isoelectric point, then it's going to be positive and negative. Because it's kind of in the transition zone. That's pretty much it on that. Have you guys had any more questions on it, or?
In question 7, it also asks like which is most likely to be found in the interior of a globular protein. So.
Yep, that one's a very tricky question.
And like.
Yeah.
Because like they're globular and fibrous, so like.
Yeah, so fibrous is more of a long chain, and then globular is where that chain kind of wraps in on itself to make a blob. So what causes it to wrap in on itself is the side chains that are hanging off of the amino acids. And essentially, a lot of the side chains that are in the center of the globular protein are hydrophobic, meaning afraid of water, being nonpolar. So amino acids that have the most nonpolar side chains are going to be found in the center of a globular protein because it's furthest away from a hydrophilic environment, which the human body, life organisms are hydrophilic environments, so they have a lot of water present. So the nonpolar amino acids are going to be far away into the center of a globular protein. So then we would have to figure out, all right, which of the amino acids has the longest nonpolar side chains. We got leucine has a side chain 1, 2, 3 that has somewhat of a side chain. We've got glutamic acid. Glutamic acid is going to be a no because it has 2 carboxylic acids which are very polar, so that's definitely a no. Leucine's a maybe. And remember, nonpolar is going to be just carbon hydrogens. So if you have nitrogen, oxygen, or a halogen, it's going to be very polar. All right, then we got lysine, serine, and threonine. Okay, lysine looks like it's going to be a no because we got 2 amines and a carboxylic acid. We got serine, we've got an alcohol right there, plus an amine, so that's a no. Threonine, again, alcohol on the side chain, so that's a no. Oh, so that pretty much narrows it down to that one, maybe, leucine, which has just a nonpolar side chain. So the correct answer would be leucine. Most likely would be found in the center of a globular protein because it has the longest nonpolar side chain. And again, if we added a different amino acid to that group, could that change the answer? Yeah, it could. If you just have to compare the particular amino acids they're asking you. And then B, that one's not too bad. It contains a secondary alcohol group. So that's organic chemistry. And I think I spotted it right here, serine. Well, actually, no, that looks primary, doesn't it? So that wouldn't be it. Oh, but threonine, this one has an alcohol coming off of a carbon that's between 2 carbons, being secondary. Whereas serine, it's hanging off of a carbon that's attached to just 1 carbon. So this is a primary alcohol, and this is a secondary alcohol. So that's the nice thing about doing biochemistry is it reviews those organic chemistry concepts. Which doesn't matter anymore because we're not taking a test. You guys feel good about that, or?
No.
No? It is a bummer because the test would have been an opportunity to kind of show what you know. But so.
Look like that's all we've been doing these past
Yeah.
poo-poo.
All right, so thin layer chromatography. First step you need to do is take your protein sample and add a diluted hydrochloric acid, preferably warm, and this is to break down the peptide bonds that are basically making the protein, connecting all the amino acids to form this protein.
What do you add to it?
You add some sort of dilute acid, so typically hydrochloric acid, and what that does is it hydrolyzes the protein, which is a reverse condensation reaction, because when we did organic chemistry we made amides. A protein is essentially an amide. It's a peptide bond. It's when you take an amine and a carboxylic acid and you combine them to form an amide plus water. And that's a peptide bond, and that's a protein. So by adding the acid, it reverses that, and it adds, essentially, you got to do it in a dilute acid solution so that there is water available, and you're adding water to that peptide bond so that it breaks apart to form the amino acid and carboxylic acid.
So would we say that it's denatured?
Well,
Happening?
you're more than denaturing it, you're destroying it. So, denaturing is pretty much unraveling. It's disturbing the intermolecular attractions of the side chains that create the structure, the globular structure of the protein. Whereas this is basically splicing the protein up so that you have the individual amino acids that form it. So it's denaturing on steroids, pretty much. You then, amino acid, you take the various amino acids and you spot them on the paper. You then put them into, in the lab it says, I believe, a beaker that is filled with your solvent. It was mostly water with a little bit of sodium chloride. And I think I did it with you guys earlier in the year where we used the chromatography columns that work a lot better. And then, of course, actual thin layer chromatography paper works better than the filter paper. The acids are going to separate because of the different solubilities. So the amino acids that are more polar are going to dissolve more readily in the sodium chloride solution and therefore be able to travel further. And size does matter. If they're both comparably similar solubilities, the one that's smaller will travel further. So it's about solubility and size that determines how far these amino acids will travel on the chromatography paper. You then let it dry, and then you spray it with an anhydrous solution, and it's going to turn the proteins or the amino acids purple. And if you get it on your hands, it turns your hands purple for like a couple days. And the last step is you compare the locations of the known amino acids with your unknown sample, and you find the RF value, the distance traveled. And that's essentially thin layer chromatography, which is one type of protein separation or identification technique.
33. Or
Page-
oil.
Hmm?
Well, yeah, but like it wanted us to reference this.
Yep.
Like,
So the main answer to that, why do you not want to touch thin layer chromatography, is because you've got proteins on your hands. Skin cells are made of proteins, and they'll get attached to the paper essentially, and then create a false reading, because the ninhydrin will identify it as protein, and it'll be this big purple splotch that has nothing to do with your experiment. Why they would want you to reference the packet, I don't know. Was that in the lab?
No, page 700.
Page 700.
No, 110.
Oh, page 710.
Yeah, 710. When carrying out paper chromatography on a sample of hydrolyzed protein.
Hydrolyzed, yeah.
Explain why it is important to avoid handling the paper, and then make reference to section 33 in the IB data booklet. Answer
for
part C.
part B and C.
Sorry.
Yep. And that's your attempt to separate mixture. Okay. Do you want to go over B and C?
Yeah, I didn't know how
Yeah.
to do those.
So 13 is just basically talking about there's proteins on your hands and you don't want to touch the thin layer chromatography because you'd get messed up data because of that. B, explain why in gel electrophoresis the amino acid isoleucine migrates towards the anode at high pH and towards the cathode at low pH. Okay, where is that? Alright, we got, and this is isoleucine. Leucine has an isoelectric point of 6. So isoelectric point, pH is 6. And migrates towards the anode at high pH and towards the cathode at low pH. So let's say we are, let me just write a quick sketch of it. Alright, so this is not isoleucine, it's just a generic amino acid. I got an amine, I got a carboxylic acid. And let's do high pH. And high pH would be, let's just say, above 6. So high pH means what type of environment? H plus wise.
Low H plus.
Low H plus. So therefore, the amino acid is going to give H to the environment, and it's going to form negative charge. And let's do an example of low pH. This is high H plus environment. Technically we put in brackets. So low pH is going to be less than 6. And that means high environment H plus. We're going to go into the amino acid, and it's going to attach to the amine, form positive. And the question was, explain why in gel electrophoresis amino acid isoleucine migrates towards the anode at high pH. So we got high pH, and it's negatively charged, and it goes to anode. Isn't the anode typically negatively charged, and the cathode positively charged?
Yeah.
Yes. So the tricky thing about this is gel electrophoresis, and what we're doing is we are adding a current to reverse that flow. So in gel electrophoresis, this is important to know, cathode is negative, anode is positive. So it's opposite because we're putting a current into it, reversing the natural flow. And if we didn't put a current into it, nothing would happen. So if we have a negatively charged entity, typically it would go to the cathode. But since we're reversing it by putting a current into this gel electrophoresis device, it's actually going to the anode. So high pH environment goes to the anode, whereas a low pH environment goes to the cathode. Questions on that? So the 4 different structures of proteins. Primary structure is the sequence of amino acid, just the chain. Secondary is Anyone remember what secondary is?
The folding of the chain due to hydrogen bonding.
You got it. So the
Gosh, my mic was muted and I said it first!
Nice.
Hello, you people.
So you got the alpha helix and the beta plata sheets. So, and it has to deal with the side chain interactions, the hydrogen bonding. And then the tertiary is the folding based off of the side chain interaction. Oh, and you have, there's 4 main types of side chain interaction that create this folding and this globular structure. Number 1 is H bonding. You then have disulfide links or salt bridges. And then you have London dispersion forces or van der Waals forces, which is just the nonpolar interaction. And then the last one is dipole-dipole, that's not hydrogen bonding, or you got various other ionic bonds that aren't disulfide links. So those are the 4 different ones: H bonding, disulfide links, dispersion forces, and then ionic/dipole-dipole forces. Those are all the forces that create the tertiary structure, the folding of the protein. And then
in the book, it said hydrophobic interaction.
Hydrophobic interaction, yes. So that would be nonpolar. So like the London,
nonpolar one.
the nonpolar, yep. So if it's nonpolar, it's going to create hydrophobic interactions. And then the last one is quaternary structure, which is the interactions of these tertiary structures with other tertiary structures based off of the intermolecular forces and the attractions. And that is it. Oh, alpha helix and beta plata sheet. The difference between those is intramolecular forces. So basically the pattern of the amino acids is going to create the spiral formation, whereas beta plata sheet, it's the attraction between the side chains. So if there are side chain attractions, it's going to fold over on itself to create the beta plata sheet folding. And if you don't have those side chains, then it's going to go into the spiral staircase alpha helix.